Yeah, is it visible, my slides? Hello?
Yes, sir. Yes, yes.
Yes, sir.
Okay. So, good afternoon, everybody. And I thank the organizer of this Healthcare World Oncology Summit, the organizers of this meeting. And today I would be talking on this topic of this, that screening, HPV vaccine, and innovative therapy of cervical cancer, the opportunity and challenges. So I have many slides, but I will try to complete within time. So let me proceed. So as you all know, all the cancer specialists is a cancer meeting, and everybody knows that the cancer is now it has become the leading cause of death worldwide. And it has been in the Lancet, it has been shown in 21 countries, accumulated data by the WHO, and accounting for nearly 10 million deaths in 2020, and nearly 1 in 6 deaths. So you can see from this picture, it is that it has become the graph on the left side, that you can see that the cancer is the leading cause of death as compared to the cardiovascular, which was in top, but now cancer has become top in the rich countries. But then the middle income countries where India comes in, so there also it is catching up, and very soon it will be the top medical journal Lancet, their global survey along with WHO projected that by 2030, every second person around the world will suffer from one or the other form of the cancer. According to Globocan and the WHO, India's cancer burden will be also double in next 20 years' times. It indicates that the cancer will be epidemic in India, which will be soon maybe the cancer capital of the world. It has already become the diabetic capital of the world. So globally, cancer also looks to be epidemic. So why should India be a cancer capital of the world? So because cancer becoming epidemic in this due to the uncontrolled pollution, rampant adulteration, contamination of air, water, soil, and so what we eat from morning to evening, and what we do, and what we breathe, and what we use in our daily life, cause heavy exposure to all of us to harmful chemicals, adulterated foodstuffs, lifestyle, smoking, radiation, pesticides, antibiotics, including pathogenic bacteria and viruses, and that affect not only our health, but that is through the health of the plants, animals, and environment. This is how the three healths, the plant health, which from where we take our food and all, many of the vegetables and many of the things, they are heavily exposed to the pesticides and all environmental. This is, I need not to explain the environmental pollutions. And the animal health, we take the animal food and all from those, and it affects our human genome or human health, and that's why we develop the various diseases. But then cancer is a genetic disease, not that it is hereditary, but in the sense that the most cancer arise due to the abnormalities in the expression of a specific genes associated with the cell proliferations and growth and their cell differentiation, because the cancer is nothing but the uncontrolled cell proliferation. So this cell proliferation or cell division is controlled by certain genes, which are, if at all, affected by these agents, then it will be a mutations or alterations, and that leads to the abnormal growth. But about 20% of the cancers, also human cancers, are caused by infectious agents. And that there are many, hepatitis B virus, herpes virus, and Helicobacter pylori, even bacteria. But what is today I'll be discussing the human papillomavirus. I think earlier session many people have just talked about the cervical cancer and HPV. So this is my area, and I am the person who introduced or the pioneered this work in India, and I worked with the pioneer who got the Nobel Prize. He is my guru, Professor Joausen from German Cancer Institute. So I have spent almost 10 years there, and then I started this research in India, and then it is now well established that the infection of human papillomavirus causes cervical cancer in women, and pathogenesis of this cervical cancer closely linked with the HPV infections. It is its interaction with the cervical epithelial cells or the reserve cells, specifically at the transformation zone, which is the squamocolumnar junction, which is mentioned. You can see it in the middle, which is colored, that the upper is the endocervix, is the ectocervix below, but then it is the squamous junction. It is called transformation zone. The new cells or the reserve cells are being produced there, and there is also stem cells. So this is I'll come with the end of these stem cells, that why these innovative drugs that we have developed. So that is against the cancer stem cells. So this is if there is an infection, so the HPV infection occurs in this squamous junction. So preferential physical interaction with HPV with the squamous junction, with the T zones, with stem cell properties, they are affected, and that leads to the tumorigenesis. But just infection doesn't mean that it will cause cancer. HPV research, that persistent infection of the HPV is important. That about almost 100% of the cancer we have shown through national mapping of the HPV and several publications, that the cancer lesions occur due to the HPV, HR-HPV infection, the high-risk HPV infection. There is around 15 high-risk types of HPVs and 10 to 12 low-risk HPVs, which are mainly cause the cervical lesion, condylomata acuminata, or the genital warts and all these warts. So cervical cancer, the statistics, 1,30,000, 30,000 or 35,000 new cases every year, and death about 73,000 deaths occurs. So that is the picture, that every one out of every two women die of cervical cancer in India. Still, though it has become the third or fourth position after the breast cancer, which has taken over the cervical cancer, more than 70 or 80% of the women certainly will get infections in their lifetime. But 90% of these cases, this HPV infection clears within 2 to 3 months' time because of their own immunity. But it takes about 10 to 15 years or 20 years to develop this malignancy, and it has well-defined precancerous and cancerous stages. So it's given a unique opportunity to understand and to screen and identify these early lesions and dysplasia or dysplastic or early precancerous lesions, and they can be well treated. So cervical cancer could be cured if it is detected early. So these are the classification, Bethesda classification, CIN classification, and WHO classification. So LCL, HCLs, and then severe, mild dysplasia, moderate dysplasia, severe dysplasia, carcinosity, and CIN 1, 2. They have a similarity, just to show that everybody knows, those who are clinicians. And this is a cervical cancer epithelium, that the HPV infection, then it comes to precancer, then to the early cancer, then precancer. And it takes almost 10 to 15 years or 20 years even. So there is ample time to identify that. But since in spite of that, we don't have a very poor screening program, and because of the trained personnel and the government initiative or the public initiative or political will, the HPV is a necessary cause of cervical cancer. That is 99%. We have shown that 100% of the cancers. But this is a genome, not to talk about anything going at the molecular, but it has a genome that is E6 and E7 are the early genes which are responsible for the tumorigenesis, called the cancer viral oncogenes, and the L1 and L2 are the capsid proteins, the principal and the major capsid proteins. But major of the vaccines are produced from L1 region. It is important that we have the first vaccine, though HBV vaccine was there, but then against the cancer, that is against the HBV, hepatitis B virus, but then it is against a cervical cancer, against the HPV 16 and 18, which is the major agent that 80% of the cancer are caused by due to HPV 16 and 18 high risk HPV, while the 6 and 11 cause the worst and Panellomata acuminata. So this is that the E6 and E7 are genes which are generally it reacts with the two tumor suppressor genes. So this is repeat of the things what are other low risk types in detail, and high risk type 16 and 18 is mainly counts for the almost 80% of the cervical cancer, but then others 31, 33, 45, 39, 35, 52, and there are several others, 13 types are there. But it is just not to bother about whole the cartoon. But what is that E6 and E7 are the viral oncogene. E6 binds with the p53 tumor suppressor gene, which and it gets degraded through ubiquitin pathways, and the E7 the p53 is degraded, whereas another tumor suppressor gene RB, retinoblastoma susceptibility gene, which causes the retinal cancer or the eye cancer in the children. But then this protein binds with the E7, and E7 is degraded. So RB is degraded. So two major tumor suppressor gene, which particularly RB helps in the uncontrolled cell proliferation. So that's is degraded. So there is no the RB is available or the two tumor substance absent. So that's why it leads to the carcinogenesis. So this is I don't go into the detail of this. This is a description of this what I just now described about the genome. But the presence of HPV, high risk HPV, indicates susceptibility to the development of cancer. But it is not that it will immediately develop the cancer. It has to persist for a year or two, and then it gets incorporated into the host cell genome. That's why persistence of high risk HPV infection and its integration into is the critical step in the development of the cervical cancer. So there is a tumor disease transformation only occurs when there is integration. So cervical cancer can be prevented by cervical cancer that already some speakers have already talked, the screening and prophylactic vaccination. But the lack of adequate health infrastructure and implementation of these programs, as I told, the political will and the government initiative is not there. So there is no screening yet, except some few organizations or few medical hospitals, medical colleges and hospitals where this facility is available and restricted. And though pap smear is very easy and the visual inspection is easy, but it is not available elsewhere. But most of the infection is generally automatically cleared by the immune system, but 10 to 12% persist and they develop the cancer. So human papillomavirus cancer, it is curable. So this is the biological behavior or the natural history of the disease. So initially it can be given before infection. You can give the prophylactic vaccines. So these two vaccines and 3 vaccines are available rather. The Gardasil, the quadrivalent and the nonavalent, 9-valent Gardasil, and the bivalent Cervarix. And that can be given in the early 9 to 26 years or 9 to 19 to 26, or it may be going to 46 years, but you need to test like SBSE, you get the hepatitis B virus infections. So vaccine, so you have to get tested. If you are negative, then it can be given the vaccines. So this is, I will not go into the detail. And you know that this is several factors, particularly the earliest marriage in India. The major of the cancer is the poor women's cancer, those who are in the village side or the countryside. So this 18 years, now it has become marriages is 21 years, but it was earlier 18 years. That was because of our study. I was in ICMR in 35 years. So there, when in director, I became director. So that time, this I published and shown in the Lancet that the 18 years is the earliest marriage. The below 18 years, they are more susceptible for HPV infection and high incidence of cervical cancer. So that is there. There are 12-year study and we showed that in the HPV detection. So after that, it was introduced in the health ministry that it will be 18 years, and later on it has become 20 years. Then multiple partners, poor genital hygiene, pregnancy, number of pregnancy, and many of the smoking and all these factors, ethnicity and religious dietary factors. So what happened is these Lancets and the WHO has answered that there is a paper in the Lancet, and there we said that towards global elimination of cervical cancer in all groups of women, particularly in the North and Southeast Asia, Asian countries, that there is a formula: 90%, 70%, and 90%. 90% the vaccination, the girls, the adolescent girls can be vaccinated 90%, and at least 70% to 80% can be screening can be done at the age of at least 35 years. At least one time screening. That is the Sankarnaran has already shown in the Tata Memorial study in the Barsi, the rural registry, that one-time screening can bring down the 50% of the cancer downstaging can be done. So that's why the one-time screening or 2 times at 45 years can be screening, so that by 2030, the goal of WHO that whole world can be eliminated with the cervical cancer, and those who are positive, they can be 90% of them can be treated. So these are the diagnostic, early detection, pap test, cytology, colposcopy, and BELI, visual inspections, and HPV DNA test. That is the most reliable, and hybrid capture 2 or CARE HPV. But there are also opportunity. That's because this takes almost 1-2 decades to appear the cancer. So any type of elimination, the preventive measures can be taken. There are herbal, there have been clinical trials. We have introduced several clinical trials which have been done, and these are references have been given. So like Artho Basu, and then the Shukla et al. These are International Journal of Cancer Research and Clinical Cancer Research. So there are several the herbal products can be used for 4 weeks plus minus the menstruation period, and topically, and that can when the HPV infection occurs, that can be easily removed or eliminated before they get integrated and persist for a longer time. So early detection by pap test. These are simple the cytology that it shows that the right side picture, you can say the LCL, but as HPV infection showing the coilocytes. That is a specific structure. It is because of HPV infection. Left side is the HCL, which is showing the precancerous lesions of the nuclear deformations. The HPV DNA testing technique is important and essential for the early detection of the high-risk populations for triaging the CIN lesions. So the dry papers are there. For these easy techniques we have introduced for HPV, that is published in the Lancet in the urine. You can take from home, you can send the urine, and HPV detection can be done. This is 100% correlated, and there are easy method of just need not to extract DNA, just boil the cell pellet, and then in that tube itself, you can use the PCR mix, and then you can have the PCR data within a 1 hour time. Or it can be collected in dry paper smear. So these are all published things, but urine method is a very excellent method and easily non-invasive method. The women against gender, the HPV vaccine was introduced in 2006, and that is Gardasil, the quadrivalent, nonavalent later on, and it has been done also study. We have done that the Tata Memorial study has shown that with the Sankarnaran that one time the 2 doses can be sufficient instead of 3 doses of these vaccines, the 0 to 1 month or 2 months and 6 months doses, the booster doses, Cervarix, bivalent, and these are the dose schedule, but it has come to 2 doses, and target population is 9 to 14 years initially, but it can go to 9 to 26 years before marriage, or it can go to 46 years still if you are not infected, if you are found to be tested non-positive, positive HPV negative, you can take this vaccine. Acceptance by the parents. There are a lot of ethical issues, the religious issues, cultural issues, but HPV vaccine in this scenario. So I'll take just 2-3 minutes. So total, this is a study done in AIMS by Nirja Bhatla. So she said that this is a Cervarix, and it has been 330 populations they did it, and she showed that this is very well tolerated and immunogenic, highly immunogenic, and compliance was 97%. So, but the problem came when it was in 2009 9 to 10, and when this path from it introduced that the HPV vaccine, the immunogenicity testing in two places, Khammam district of the Andhra Pradesh and the Vadodara district in Gujarat. And that time it had, there are 6 girls, there are given to the school girls the tribal area, but then 6 girls died of this, but they are unrelated to, proved to be later, they're unrelated to the vaccination. But all the people, all the women activists and other activists, they said that there are irregularities in the conducting the vaccine trial, and so this matter is to the court, and this is hanging in the court still date. But that's why there are more than 100 countries has introduced the vaccine, and in UK and US, but in India, which is the top cervical cancer incidence, but we have not yet been able to put in our national vaccine immunization program yet. That is very shame, and there is no political will now. Government attempt, or there is no. Everybody is very silent. But though the WHO has called for the elimination of cervical cancer, it is more appropriate for the India. But WHO has supported the government of Punjab and for HPV vaccination program in district of Bhatinda and Mansa in 2016. And Delhi has taken this HPV vaccine program introduced, and in the public health program in the school children. So this has the 2 states has taken, but it has not become the. But one can get privately in the private clinics. You can have the vaccination. Those who are educated parents, they generally go and get their daughters vaccinated. So this is available, and there are new 2nd generation vaccine, the DNA vaccine, 9-valent vaccine. I told you there are Serum Institutes of India, Pune, but this has not come into the vaccine cost. Though we have come into mark, and the GSK has bring down the price. But the Bharat Biotech, we don't have our indigenous vaccines till they are under productions. We have also, when I was director of ICPO, which is an ICPR now of the ICMR, I started the vaccine chimeric DNA vaccines. This was vaccine construct has been prepared. The time is less. I will not just go into the detail. We made the vaccine construct, and we have seen the serological test developed to detection of the HPV, anti-HPV 16 antibody, because we have had the Indian variant HPV 16 vaccines construct, and that is the beauty of that. And it was observed in the that they found that the V16 the variants was the most immunogenic, and they are so it seems to be 2-fold higher immunogenic than other vaccine the variants. So it is in the animal trial, but if it is expected because of COVID, it is delayed too much. So it may be 2-3 years time. It will be going to the human trial, and we will be able to bring that vaccine. This is a chimeric vaccine, both 16 and 18, and it is a DNA vaccine. So once given, 1 dose is sufficient. So just 2 minutes. So there are all these herbal products are there. So curcumin and this is the clearance. But novel drug discovery, that is the last point. That is 2-3 slides. The folate receptor mediated targeted delivery, the curcumin, and the drugs, the chemotherapy drugs to the cancer cell, along with the cancer drugs, developing a triple conjugate. It's called triple conjugate. The drug, there is no drug available to target the cancer stem cell, which are responsible for the relapse or recurrence of the cancer, all types of cancer. We are doing in other cancers also, breast cancer also, but I am presenting here the targeted delivery, the curcumin to cancer and cancer stem cell by triple conjugate drugs, where we have the curcumin and the triple conjugate, where we have the drugs as well as folate receptor. So what we have shown, that we have not much time, that why these cancer stem cells are resistant to the radiotherapy and chemotherapy, because ABCG2, that they have the protein, they produce this protein, ABCG2 protein, that protein reflects out these cytotoxic drugs and radiations. And that's why they are protected and they remain in one corner, 1 or 2%. And after 5 years, 3 years, 5 years, they repopulate and they become aggressive. So this is the triple conjugate. We have the doxorubicin, and we have made the linker with the curcumin, with the doxorubicin, and the folate receptor. It is delivery. So we'll not go into detail. It is delivery because it is well-established system for the targeted delivery, the university drug to the through siRNA, protein toxins, and anything. So the folate uptake follows a natural vitamin uptake, endocytosis pathways. The folic acid being used as a targeting ligand. Folic acid can retain. So it is the folic acid can be conjugated with the carboxyl group, with the hydroxyl group of the curcumin, with the ester bond and the ligand that goes to the inside, like this picture, the vitamin uptake procedure. So you have the high receptor expression in the cell membrane, and from there the investigation is the caveoli or clathrin pits. It gets into the cell cytosol, and then in the cytosol pH, this will break down, and the bond will break down, ester bond, and they will release the curcumin and the doxorubicin, and the curcumin will sensitize the cancer stem cells. So that has been already been shown by us and published, and then the doxorubicin can kill those cancer cells, cancer stem cells. So we have shown that it is known that the folate receptor is not normal cell expressed. So it is only expressed by the cancer cells and cancer stem cell, barring these few lung, placenta, and thyroid, and fetal lungs, and trachea, and kidney. Besides that, other cells do not express folate receptor. So it will go to only to the cancer cell. So we have the non-toxic small molecular weight curcumin folic drug conjugate, which can especially go to the taken up by the cancer cells via receptor mediated internalization due to overexpression of the high affinity folate receptor, will yield the curcumin, and upon enzymatic cleavage of the ester bond. This approach will improve the bioavailability of the, we have made the little modification to bioavailability of the curcumin and targeted delivery of the curcumin along with the anti-cancer drug, also prevent the non-specific metabolic inactivation. So identification and of therapeutic target and development of the conjugate drugs for cervical cancer for relapse-free survival of the targeting cancer stem cell, which has the major principle, the main principle of the factor of the cancer recurrence. So this is the concluding slide, last slide. The novel curcumin folate drug conjugate may open the excellent, because we have already in clinical trial in animals, the possibility, and it has been patented for a medical innovation and targeted delivery of drugs on the chemo-radio-resistant cancer stem cell in order to make the cancer treatment most effective and relapse-free. Together, HPV vaccine and screening, and above this triple conjugate drug for the relapse-free survival, if we go ahead with that, and that will make the our cancer treatment most effective, and we can expect by 2030, if not 2030, by 2050, the India should be cancer-free, cervical cancer-free, like our polio and smallpox. But screening must be done, and the HPV vaccination should be 70 to 80% to 90% should be done, and if detected, they should be treated 90%. So this is all about I wanted to share with you.
Thank you so much,
so this
Dr.
is,
Das.
I have